Hello everyone. Welcome back to my channel XY Biology Academy. In today's experiment, we'll learn one of the most important techniques used in microbiology, culture transfer technique, also known as subculturing. Subculturing is the process of transferring microorganisms from one culture medium to another under aseptic conditions. It is routinely used to maintain pure cultures, prepare stock cultures, and perform various microbiological tests. Since microorganisms are naturally present in the air, on laboratory benches, and even on our hands and equipment, every transfer must be carried out carefully using aseptic techniques to prevent contamination and obtain reliable experimental results. The aim of this experiment is to learn the correct aseptic technique for transferring microorganisms from one culture medium to another without introducing contamination. The principle behind this experiment is simple. Microorganisms are transferred from an existing culture, known as the stock culture, into a fresh sterile culture medium. During the transfer, every instrument and every opening that comes into contact with the culture must remain sterile. This prevents unwanted microorganisms from entering the culture and ensures that only the desired organism grows in the new medium. Before we begin the procedure, let's quickly understand the complete workflow of this experiment. First, we'll prepare and label all sterile culture tubes. Next, we'll sterilize the inoculating loop or needle. We'll then collect microorganisms from the stock culture under aseptic conditions and transfer them into fresh sterile media. After the transfer, we'll sterilize the equipment again, close the culture tubes properly, and finally incubate the inoculated media to allow microbial growth. Now that we understand the overall process, let's look at the materials required. For this experiment, we need the following materials: stock culture of microorganisms, sterile nutrient broth, nutrient agar slants, nutrient agar deep tubes, inoculating loop, inoculating needle, Bunsen burner or micro-incinerator, marker for labeling, test tube rack, incubator. Now we understand the procedure of this experiment step by step. Step 1. Label the tubes. The first step is labeling all sterile media tubes with the name of the microorganism and your initials. Proper labeling helps prevent confusion and ensures accurate identification throughout the experiment. Next, hold the stock culture tube and the sterile medium tube together in one hand, forming a V shape. This position allows both tubes to be handled efficiently while minimizing exposure to airborne contaminants. Step 3. Sterilize the loop or needle. Now sterilize the inoculating loop or needle by heating it in the hottest part of the Bunsen burner flame until it becomes red hot. This destroys any microorganisms present on the instrument. Allow it to cool for about 10 to 20 seconds before touching the culture. Step 4. Remove the tube caps. Carefully remove the caps using the fingers of the same hand that holds the loop. Never place the caps on the laboratory bench, as this can contaminate them. Keep the caps in your hand throughout the transfer process. Step 5. Flame the tube mouths. Briefly flame the mouths of both tubes 2 or 3 times. This helps reduce contamination by killing microorganisms around the opening and creating an outward flow of warm air. Step 6. Collect the inoculum. After the loop has cooled, collect a small amount of inoculum from the stock culture. If using a broth culture, take a loopful of broth. If using an agar slant, gently touch the microbial growth without damaging the agar surface. Step 7. Transfer the culture. Now transfer the inoculum into the sterile medium. For a broth-to-broth transfer, gently mix the loop inside the broth. For a broth-to-slant transfer, streak the agar surface from the base toward the top using a straight or zigzag motion. For a slant-to-agar deep transfer, use a straight inoculating needle to make one vertical stab into the center of the agar. This method is known as stab inoculation. Step 8. Flame again and replace the caps. After inoculation, remove the loop carefully and flame the mouths of both tubes once again. Immediately replace each cap onto its original tube to maintain sterility. Step 9. Re-sterilize the loop. Finally, sterilize the inoculating loop or needle again until it becomes red hot. This destroys any remaining microorganisms and prepares the instrument for future use. Once all transfers have been completed, incubate the inoculated cultures at 25 degrees Celsius for 24 to 48 hours. During incubation, the transferred microorganisms will grow in the fresh culture media, allowing successful subculturing to be confirmed. After incubating the inoculated cultures at 25 degrees Celsius for 24 to 48 hours, microbial growth should be visible in all the freshly inoculated media. In the nutrient broth, successful growth is indicated by turbidity, meaning the broth appears cloudy due to the multiplication of microorganisms. On the nutrient agar slant, visible colonies or a continuous streak of microbial growth should appear along the line of inoculation. In the nutrient agar deep, growth should be observed along the stab line made by the inoculating needle. If aseptic technique has been followed correctly, only the intended microorganism will grow, and there will be no unexpected colonies, unusual colors, or growth patterns. This confirms that the culture transfer was successful and the culture remains pure, free from contamination. In conclusion, the culture transfer technique is one of the most fundamental skills in microbiology laboratories. By following proper aseptic procedures, sterilizing instruments, flaming culture tubes, and handling cultures carefully, we can successfully transfer microorganisms while maintaining pure cultures and preventing contamination. Mastering this technique is essential for microbial isolation, identification, culture maintenance, and many advanced microbiological experiments.